Respected Dean Sir, respected Seminar in Charge, Major Advisor, Member of Advisory Committee, Professors, and my colleagues. Today I am going to present my pre-thesis seminar on studies on bacterial biodegradation of low-density polyethylene. The plastic pollution is a major concern. It not only affects the environment but also affects the animals. The animal were died due to the plastic ingestion, and out of them, the LDPE, low density polyethylene, is mostly used, and the degradation of this polythene was not very easy. That scientists were tried to degrade it, but the recycling and landfilling, land dumping, incineration is another technique can't be helped. So the degradation with the help of different means are required. Hence, looking to that, my objectives during my studies were isolation and identification of low density polyethylene degrading bacteria to determine the biodegradability of low density polyethylene, comparison of degradation ability of different biodegrading bacteria. For that, the material and methods which was required, the work was carried out at the Department of Veterinary Microbiology, College of Veterinary Science and Animal Husbandry, Mahu, to fulfill first objective, that was the isolation and identification of low density polyethylene degrading bacteria. The isolation of bacteria was performed by using LDPE, that was selected on the basis of Mariotti et al. 2019, and that was measured by micrometer caliper. Then further it was treated with benzene for 30 minutes to remove any plasticizer and coloring agent. Then this LDPE were used for the preparation of LDPE films and LDPE powder. The LDPE films were prepared according to Lee and co-workers 1991 and Yao and co-workers 2022, that were cut into 1.5 into 1.5 centimeter square size for used in the MSM agar, and 1.5 into 1.5 centimeter square, then 1.5 centimeter diameter circle, 2 centimeter diameter circle, and 1.5 centimeter triangles were used for the MSM broth. Then the UV exposure was given to this LDPE films before uh, uh, after the cutting, and the FTIR was analyzed before and after the UV exposure. These LDPE films were then weighted separately and then stored into the glass desiccator. Likewise, the preparation of LDPE powder was done according to Nanda and co-workers 2010 and Bhatia and co-workers 2014, and then again the UV exposure was given at 365 nanometer at for the 48 hours. Then this powder were again stored into a closed container. The LDPE film and LDPE powder were used as a substrate, as a carbon source for the growth of the bacteria. Now the samples were collected from the veterinary clinical complex and instructional livestock farm complex, Veterinary College Mahu. These samples were fecal sample from cattle, buffalo, goat, horse, poultry, pig, and dog, and the sewage water, dumping ground soil, garden soil were collected from site of dumping area and polluted area in and around Mahu. So this is a table showing the different type of samples were collected from different latitude and longitude, and they were given the lab numbers. In the whole study, mineral salt medium were used, and that was prepared according to Nademo and co-workers 2023, and other media like nutrient agar, urea botany glycerol broth, and Simon citrate agar, glucose phosphate broth, peptone water were also used. MSM broth and MSM agar were used according to the experiment, like MSM broth with 0.2% LDP powder were used for enrichment of bacteria. MSM agar with 4 LDP films and 0.2% LDP powder were used for isolation of bacteria. MSM agar and MSM broth containing 4 LDP films were used for biodegradation study. Then MSM agar with 0.2% polyethylene glycol were used for the clear zone analysis. Now comes to the isolation of LDPE degrading bacteria. For that, the MSM broth were used. In that, 1 gram of sample were inoculated, and 0.2% LDPE powder was present inside. It was incubated at 30 days at 37 degrees centigrade at 120 rpm. Then further, after 30 days, gram staining were performed for the presence of bacteria. After 30 days of enrichment, 1 ml of this broth were inoculated onto the MSM agar plate containing 0.2% LDPE powder and 1.5 into 1.5 centimeter square LDPE films. They will be incubated aerobically for 60 days, and after 60 days, the gross morphological colonies were selected and then transferred to the nutrient agar and then again inoculated for 1 to 2 days at 37 degrees centigrade. Further, these isolates were identified microscopically and biochemically. Microscopically, gram-positive and gram-negative bacteria were observed, and biochemically, the described procedure by Barrow and Feltham 1993, Malik 1995, and Coley and co-workers 1996 were used. After the isolation of LDPE degrading bacteria, they will be screened by clear zone analysis according to Rana and Rana 2020. For the screening, MSM agar containing 0.2% polyethylene glycol were used. They will be aerobically incubated at 37 degrees centigrade for 14 days. After 14 days of incubation, these plates were stained with 0.1% Comasi Brilliant Blue solution and de-stained with the 40% methanol and 10% acetic acid to visualize clear zone. Those bacteria which were forming clear zone, they will be selected for further biodegradation study. Then identification of the bacteria were done by BD Phoenix M50 automated and identification and susceptibility system, and further they will be identified by sequencing by using 16S rRNA gene with the universal forward and reverse primer. By sending these isolates to the Barcode Bioscience, Bangalore, Karnataka, India for single sequencing. Then these sequences were submitted to the NCBI by using BioEdit software, and the phylogenetic analysis was prepared by using Mega 11 software using neighbor joining method considering 1000 bootstrap values. Now comes to the results of objective first, and that was presence of gram positive and gram negative bacteria into this plate. Further, they will be incubated for 60 days, and 28 isolates will be isolated. Out of them, 12 isolates were gram positive and 16 isolates were found gram negative. These isolates were also given the lab numbers. This table showing the 28 colonies. They will be characterized biochemically by catalase test, oxidase test, and IMVIC test. The results of catalase test is showing the presence of bubbles. Then results of oxidase test is showing the purple color in this plate as a positive test. Then ring of red color over the surface indicated positive test of indole test. Then red color of methyl test is showing the positive result. Ring of bright red color on the surface of broth showing the positive test for the Vegas box here, and change in the color from green to blue is indicated for positive test of satellite utilization. Further, this bacteria, this 28 isolates were inoculated onto the MSM agar containing 0.2% polyethylene glycol, and the result showed that no clearance as a negative, then minimal clearance as positive, then moderate clearance as double positive, and the maximum clearance as triple positive. The maximum clearance was seen by 5 isolates, and they will be shown in this table. These 5 isolates were identified by BD-Phoenix M50 automated identification and susceptibility testing system, and further they will be characterized by sequencing, and the accession number is showing in this table. Phylogenetically, these isolates were characterized. Phylogenetically, these isolates were found homologous to the Staphylococcus glossy. The M4 isolate were homologous to the Cocorea rosea strain. Then P9 isolate was homologous to the Pseudomonas aeruginosa, and S1 was homologous to Cellulose microbium cellulans, and S2 was homologous to Leuconostoc mesenteridis subspecies cremoris, and the same is showing in this table. The Staphylococcus was uh, grown on the as seen on the LDPE film as a white circular colony that is showing in this plate. The plate showing the presence of Staphylococcus clausi, and the same is showing as in the ground staining. This plate is showing Cocoria rosea on the surface of LDPE film, and the same is presenting into the nutrient agar plate with their gram staining. This plate is showing Pseudomonas aeruginosa on the surface of LDPE film, and the same is presenting on the nutrient agar, showing the greenish pigment and the presence of gram-negative organism in the gram staining. The cellulose microbiome cellulans were presented here as a rhizoid colony on the surface of LDPE film, and the same is presenting on the nutrient agar, and the gram staining were shown as gram-negative rods. The presence of a colony of Leuconostoc mesenteridis on the LDPE film, and the same is presented in these plates, showing the colonies in the nutrient agar and the gram-positive cocci of the Leuconostoc. These colonies are pigmented, and the different pigments are showing in this plate. Now comes to the conclusion. Staphylococcus clausi M1, Cocoria rosea M4, Pseudomonas aeruginosa P9, Cellulose microbiome cellulans S1, and Leuconostoc mesenteridis S2, isolated from cattle feces, sewage water, poultry droppings, cattle feces, and buffalo feces respectively, were all effective in degrading low-density polyethylene. Now to fulfill objective second, that was to determine the biodegradability of low-density polyethylene, the experimental setup was conducted. In that setup, 6 petri dishes were arranged with the 4 LDPE films, and this group of 6 petri dish containing 0.5 McFarland inoculum, and S1, S2, M1, M4, and P9 representing the number of isolates, having one control group. So the arrangement of 6 plates was in the form that they will be incubated for 15 days, 30 days, 45 days, and 60 days. And likewise, the broth were also incubated for the 15 days, 30 days, 45 days, and 60 days of the incubation, containing 4 LDPE films with the different sizes. Then, after different incubation periods, the films were collected for gravimetric analysis, functional group analysis, and surface analysis. The gravimetric analysis was conducted at 15 days, 30 days, 45 days, and 60 days of the incubation. Then, surface functional group analysis was conducted only at the 60th day of the incubation, and surface analysis all were also conducted at the end of 60th day of the incubation. For the gravimetric analysis, the film were collected according to Hirschhausen and Jha, and the gravimetric analysis was done by using this formula. The gravimetric analysis was done by ANOVA as described by Snedekor and Cochran. Comes to the results. At the starting, the initial weight of LDPE were taken, and further it will be incubated for 15 days, 30 days, 45 days, and 60 days, and the final weight was taken. Then the weight loss and weight loss percentage were calculated, and likewise this is for the LDPE number 1, this is for LDPE number 2, and this is for LDPE number 3. This whole table is for control group. Then this table is in the this table is for the MSM agar, and this table is for the MSM broth for the control group. So likewise, the LDPE films were analyzed gravimetrically for the weight loss. This table is showing weight loss by Staphylococcus clausi in MSM agar at different incubation period, and this is in MSM broth. This is the table showing the Cocoria rosea degradation by the Cocoria rosea in MSM agar, and this is in MSM broth. This table is representing weight loss done by Pseudomonas aeruginosa in MSM agar, and this is in MSM broth. This table is for degradation done by cellulose microbiome cellulase S1 in MSM agar, and this is in the MSM broth. This table is showing the weight loss of LDPE films in the MSM agar done by leuconostoc mesenteritis, and this is in the MSM broth. Now, the same is representing as a mean weight loss done by control and 5 isolates, and they are representing in this graph. It is showing that the highest degradation was seen by Pseudomonas aeruginosa and cellulose microbiome cellulase at the 15th day of the incubation period in MSM agar. This is the graphical presentation showing the highest degradation was done by Pseudomonas aeruginosa at the end of 30th day in the MSM agar. Likewise, this graph is at the end of 45 days, and this is the end of 60 days of the incubation, showing that Pseudomonas showed highest degradation at the end of 60th day in the MSM agar. Now, for the MSM broth, this graphical presentation shows that Pseudomonas aeruginosa and cellulose microbiome cellulase S1 showed highest degradation. Then, at the end of 30th day, it was the end of 45th day, and it was end of the 60th day of the incubation period, showing that Pseudomonas aeruginosa P9 showed highest degradation in the MSM broth. Now, the analysis was conducted, and it showed that in the MSM agar, now the analysis of weight loss reported at MSM in the MSM agar at different incubations, it shows that among the different incubations, there was a significant weight loss was observed from Staphylococcus clausi, then Pseudomonas aeruginosa, and leuconostoc mesenteritis S2, while the non-significant weight loss was reported by Cocoria rosea M4 and cellulose microbiome cellulase S1. The same is representing in this table that at the different incubation period, the significant weight loss was reported by the Staphylococcus clausi M1 in the MSM agar. And the same is representing in this graph, showing that highest degradation was reported at 60th day of the incubation period. Now, in case of the MSM broth, all isolates were showing significant weight loss during all days, and Staphylococcus M1 were presenting significant weight loss in the MSM broth at different incubation period, and the same is representing into the graph that it is showing the highest degradation at the 60th day of the incubation period. Then the Cucuria lozia M4 were presented into the MSM agar, showed the non-significant weight loss was reported at different incubation periods, and the same is representing in this graph, showing that the highest degradation was reported at 45 days of the incubation period. Cucuria rosea in the MSM broth showing the significant degradation, and the same is representing that at the 60th day of the incubation it was highest degradation reported in MSM broth. Pseudomonas aeruginosa in the MSM agar showing the significant weight loss, and the same is representing here that in the graph at 60th day of the incubation highest degradation was reported in MSM agar. In the MSM broth, Pseudomonas aeruginosa P9 showed significant weight loss, and the same is representing here at the 60th day of the incubation it was the highest. Cellulose microbiome cellulase S1 were reported in the MSM agar non-significant weight loss, and the same is representing that it was the highest at 60th day of the incubation period. In the MSM broth, cellulose microbiome cellulase S1 were reported significant weight loss, and it was the same presented here that on the 60th day the weight loss was highest. In the MSM agar, leuconostoc mesenteritis S2 were showed significant weight loss, and the same is reported that it was highest at 60th day of the incubation period. Then in MSM broth, leuconostoc mesenteritis S2 were showed significant weight loss, and the same is presented here at 60th day of the incubation it was the highest. Now comes to the conclusion. that the LDPE degradation was significantly increased over time with Staphylococcus clausi M1, Pseudomonas aeruginosa P9, Leuconostoc mesenteritis S2 in both MSM agar and MSM broth. The Cocoria rosea M4 showed maximum degradation of LDPE at 45 days of incubation in MSM agar, while it was maximum at 60 days of incubation in MSM broth. A non-significant degradation over time was reported by Cellulose microbiome cellulase S1 in MSM agar, while significant degradation was seen in MSM broth. Now, to fulfill objective third, that was a comparison of degradation ability of different biodegrading bacteria. Again, the ANOVA was uh, used as described by the Singapore and Cochrane 1994, and the results of FTIR and SAME were analyzed for the biodegradation ability of the isolates. For that, collection and observation of the LDP films were for the functional group analysis were done as per the Socrates 2004. The collection was done as described by the Ozmir et al. In 2022, and the collected LDP were sent to the sophisticated analytical instrumentation facility Indore, IIT Mumbai, Powai, Maharashtra, for further analysis of functional groups. The FTIR were compared in 3 points. First, UV untreated LDP film were compared to UV treated LDP films. Then UV treated LDP control films before incubation were compared with the UV treated LDP films control films after incubation in MSM agar and MSM broth. And third, UV treated LDP films at 0 day of incubation and then after 60th day of the incubation. Further, after analysis of the functional groups, the carbonyl index were calculated according to Albertson and co-workers 1987. If an increase or decrease in the carbonyl index was indicated for the LDPE degradation. Then, for the collection of LDPE film for the surface analysis, the collection was done as per Harshvardhan and Jha 2013, and these LDPE films were sent to Sophisticated Instrumentation Center IIT Indore and Sophisticated Analytical Instrumentation Facility IIT Mumbai for surface analysis. Any change in the surface like erosion, spit formation, crack formation, grooves on the surface were reported as a LDPE degradation. Now comes to the results. First, comparison of biodegradability of isolates in MSM agar at different incubation period. So the different isolates were compared, and it was showed that the LDPE degradation was significant at 30 days, 45 days, and 60 days of the incubation among the isolates, while it was non-significant at 15 days of the incubation in between the isolates. The same is representing here that in the MSM agar at 15th day of the incubation period, the non-significant weight loss was reported among the isolates, and the graphical representation shows that among the all isolates, Pseudomonas aeruginosa showed highest degradation in the MSM agar at 15th day of the incubation period. Then in the MSM agar at the 30th day of the incubation, all isolates reported significant weight loss, and the same is representing in the graph that Pseudomonas aeruginosa showed highest degradation among all isolates. Then in MSM agar at 45 days of the incubation period, all isolates showed significant weight loss, and it was reported that Pseudomonas aeruginosa P9 showed highest degradation. That at the end of 60 days of the incubation in the MSM agar, all isolates showed highest significant degradation, and it was the highest in Pseudomonas aeruginosa. And this is the comparative results of all degradation of LDPE done by isolates, and it was showed that the highest degradation was reported by the Pseudomonas aeruginosa at 60 days of the incubation period into the MSM agar. Now, comparison of biodegradability of isolates in the MSM broth at each incubation period. So it was reported that the significant weight loss was there at different incubation periods done by different isolates. And it was reported at 15th day of the incubation period in the MSM broth, the significant weight loss was reported by all the isolates. And it was reported same here that Pseudomonas aeruginosa P9 showed highest degradation. In the MSM broth, at 30 days of the incubation period, all mean values are found significant, and different superscript differ significantly, showed that there was significant weight loss. And the same is presenting into the graph that showing Pseudomonas aeruginosa P9 showed highest degradation at 30 days of the incubation period into the MSM broth. At 45 days of the incubation period, the significant weight loss was reported among the all isolates, and different mean values with different superscript showed significant weight loss. And the same is reporting that the Pseudomonas aeruginosa P9 showed highest degradation at 45 days of incubation period in MSM broth. At 60 days of the incubation period, all isolates show significant weight loss into the MSM broth, and these are reported same here that Pseudomonas aeruginosa P9 showed highest degradation at 60th day of the incubation period in MSM broth. The same is representing the combined representation of all the isolates with their LDPE degradation, showed that the highest degradation was reported by Pseudomonas aeruginosa at 60 days of the incubation period into the MSM broth. Now, comparison of biodegradability of isolate between the media was also done, and it was reported that a non-significant weight loss was done by control group at 15, 30, 45, and 60 days of the incubation period among the different media, that is in the MSM agar and MSM broth. Then the non-significant weight loss was reported at the 15th day of the incubation period among the media done by the different isolates. And the non-significant weight loss was reported at 30 days of the incubation period among the media by the different isolates. Again, the non-significant weight loss was reported by the different isolates, but it was significant by the leuconostoc mesenteritis S2 among the media at 45 days of the incubation period. At the 60th day of the incubation, only Cucuria rosea showed significant weight loss in between the media, and non-significant weight loss was seen by other. If we also analyzed the all incubation period at among the media by the different isolates, and it was reported non-significant. So this is the table showing at the 15th day of the incubation period, there were non-significant weight loss done by the Staphylococcus clausi among the media. This is the table for 30 days. This is for 45 days, and this is for 60 days, showing non-significant weight loss in the by the MSM by the Staphylococcus clausi M1 among the media. This graphical representation showed that Staphylococcus clausi gave the highest degradation into the MSM broth, and this was the non-significant among the media. Now, this tabular representation of the Cucuria rosea at 15 day, 45 day, then 60 day of the incubation period, and all showed non-significant weight loss in between the media. And this graphical presentation showed that it was the highest at 60 days of the incubation period in the broth, and that was non-significant. Now, this tabular representation for the Pseudomonas aeruginosa P9 at 15 days of the incubation period, and it showed non-significant weight loss at 15 days, 30 days, then 45 days, and 60 days of the incubation period, and it was found highest at 60 days of the incubation period among the media. Then this table shows the weight loss done by cellulose microbiome cellulase S1, and it showed that there was a non-significant difference done at 15 days, 30 days, 45 days, and 60 days of the incubation period done by the cellulose microbiome cellulase S1 in between media. And the same graphical representation showed that at the 60th day of the incubation period, it was the highest in MSM broth. Then leuconostoc mesenteritis showed a non-significant difference at 15th day, 30th day, while it was significant at 45 days, and non-significant again at the 60th days of the incubation period in between media. And the same is representing to the graph showed that it was the highest at 60th day of the incubation into the MSM broth. So these are the comparative weight loss of LDPE film done by the different isolate in MSM agar and MSM broth, and it showed that the Pseudomonas aeruginosa P9 reported maximum degradation into the MSM broth at 60th day of the incubation period. Now the second analysis was done for the FTIR, and it showed that the three characteristic peaks of the LDPE film before UV rays exposure, and the same is representing into the graph. Then after UV exposure, the functional group were analyzed, and the change in the intensity of absorption peaks were reported, and the same is representing into the graph, showing that the blue line without any UV exposure, and the red line showed after the change in the absorption peak in the LDPE film functional groups uh, done by the UV exposure. Now, the second analysis of the FTIR was in between control group before incubation and after incubation. The control was UV treated LDPE films before incubation, and the same control was analyzed after 60th day of the incubation, and it was reported that there was no any change in the absorption peaks. Then the third analysis was the comparison in between UV treated LDPE films before incubation and UV treated LDPE film exposed to different isolates was there, and it was reported at the end of 60th day the change in the intensity of absorption peak done by the Staphylococcus clausi M1. The same is representing into the graph, showing blue color was the control and red one was indicated that the change in the absorption peaks after end of 60 days of the incubation period into the MSM agar. Then that was in the MSM broth. Staphylococcus clausi showed the change in the intensity of absorption peak of the LDPE film at the end of 60 days of the incubation period, and the same is representing into the graph, showing blue color for control UV treated LDPE film before incubation, and UV treated LDPE film that was again treated with Staphylococcus clausi M1. Now, the change in the intensity of the absorption peak were reported by Cucuria rosea M4 after 60 days of the incubation period into the MSM agar, and the same is representing into the graph, showing the changes in the absorption peaks. Then in the MSM broth, Cucuria rosea showed change in the absorption peaks, and they were reported again into the graph. Then in MSM agar, Pseudomonas aeruginosa showed changes in the peaks of the functional group, and the same is representing that the change in the functional groups after the degradation by the Pseudomonas aeruginosa. Then in MSM broth, the change in the intensity of absorption peak were reported by Pseudomonas aeruginosa, and the same intensity change were represented in this graph. In the MSM agar, cellulose microbiome cellulans were reported change in the intensity of the absorption peak, and the same intensity were reported change in the intensity were reported into the graph. Then in MSM broth, cellulose microbiome cellulans were reported change in the intensities of absorption peaks, and the same is representing in the graph. The blue line is for UV treated LDP film before incubation, and the red one is after incubation with the cellulose microbiome cellulans at 60 days of the incubation period. Then in the MSM agar, change in the intensity of the absorption peak were reported of the LDP film. Then the same is represented into the graph. Then in the MSM broth, leuconostoc mesenteritis showed changes in the LDP film functional groups, and the same was reported in the graph. After the functional group analysis, the carbonyl index was calculated, and it was showed that the change in the carbonyl index was there before UV exposure and after UV exposure. The carbonyl index was increased from 4.824 to 12.670. So At the end of 60 days of the incubation period into the MSM agar, the change in the carbonyl index was seen, and it was decreased by different isolates. And the highest decrease of carbonyl index was seen by Pseudomonas aeruginosa. The carbonyl index was calculated for the MSM broth, and it was reported that the decreased carbonyl index was highest done by Pseudomonas aeruginosa P9. Then the percent decrease in carbonyl index were calculated according to this formula, and it was showed that the highest decrease in the carbonyl index percentage was seen by the Pseudomonas aeruginosa P9 of the UV treated LDPE film at the end of the incubation period, at the end of 60 days of the incubation period into the MSM broth. And the same is representing into the graph showing that the highest degradation and the highest percent decrease in carbonyl index was reported by the Pseudomonas aeruginosa P9 in the broth. Then the third one was the analysis of the surface for any erosion, spit formation, cracks, grooves over there after the incubation, and it was analyzed, and this plate is showing a smooth surface of the control before incubation at magnification of 2500x and 5000x. Then there was again the smooth surface were reported after end of 60 days of the incubation into the MSM agar. The smooth surface was reported at the end of 60 days of the incubation into the MSM broth. Now there were erosions on the surface were reported in the done by the Staphylococcus clausium 1 when it was removed from the MSM agar, and surface erosions with pit formation and groove formation over there when removed from the MSM broth was reported done by Staphylococcus clausium 1. The presence of bacteria on the film surface was reported from the MSM agar, and presence of bacteria on the surface of LDPE films were reported from the MSM broth. Then for the Cucuria rosea, the surface erosions in the pit formation and crack formation was there that was reported when removed from the MSM agar, and the surface erosions and groove formation was there when it was removed from the MSM broth. Then the surface erosion was reported by the Cucuria rosea M4 into the MSM broth. This plate is showing the presence of Cucuria rosea on the surface of LDPE films after removal from the MSM agar, and these are the Cucuria rosea on the surface of LDPE film after removal with the MSM broth. These are the surface erosions in the form of groove formation, pit formation, and cracks over there after removal from the MSM agar that was done by the Pseudomonas aeruginosa. And these are the pit formation on the surface of LDPE films after removal from the MSM broth. The Pseudomonas aeruginosa were reported from on the surface of LDPE film showing the crack formation over there that were removed from the MSM agar, and these cracks were shown when the LDPE films were removed from the MSM broth. The surface erosions were reported by the cellulose microbiome cellulans after removal from the MSM agar, and this surface erosion were reported after removal from the MSM broth. And the presence of bacteria, cellulose microbiome cellulans were reported on the surface of MSM of the LDPE film after removal from the MSM agar, and that were reported after removal from the MSM broth. The surface erosion was reported from done by this leuconostoc mesenteritis S2 when removed from the MSM agar, and this was from the MSM broth. The presence of pit formation and surface erosions was there. The presence of leuconostoc on the surface of on the surface of LDPE films when removal from the MSM agar, and this was the from the MSM broth. So now comes to the conclusion. The biodegradability of all isolates were reported higher in MSM broth compared to MSM agar. Pseudomonas aeruginosa showed highest LDPE degradation in the MSM agar and MSM broth, and photooxidation by UV rays enhanced biodegradation. The decrease in carbonyl index by cleavage of carbonyl bonds of LDPE was reported for by the biodegrading bacteria. Comes to the overall conclusion of my whole research work. Into the three findings are there. Staphylococcus clausi M1, Cucuria rosea M4, Pseudomonas aeruginosa P9, Cellulose microbiome cellulans S1, and Leuconostoc mesenteritis S2 isolated from cattle feces, sewage water, poultry droppings, cattle feces, and buffalo feces respectively were all effective in degrading low density polyethylene. LDPE degradation increased significantly over time. The biodegradability of all isolates were reported higher in the MSM broth than MSM agar, and the Pseudomonas aeruginosa P9 showed the highest LDPE degradation in broth media. The increase in the carbonyl index by UV rays, followed by a decrease in the carbonyl index done by bacterial action, reported biodegradation by the bacteria. Now, this is suggestions for the further work. Studies on other biotic component for isolation of LDPE degrading bacteria can be done. A study on biodegradation of other plastics, that is polyethylene terephthalate, high density polyethylene, polyvinyl chloride, polypropylene, and polystyrene can be done. Studies on bioenzymatic degradation of LDPE and other plastics can be done. A study on the plastic degradation using combined microbial consortia can be done. Expression of gene associated with plastic degrading enzymes and characterization of biodegradation associated with plastic degradation can be done. Degradation kinetic using other environmental factor like temperature, salt concentration, incubation period, growth media can be done. Thank you.